So next, we will have Dr. Raphael Trevizani join us to present Receptor Tools, TCRMatch. And then towards the end, Raphael will hand it over to Dr. Mahita Jarjapu. These are two of our bioinformatics postdocs, if I didn't mention that already. And Mahita will give a really brief update on TCRMatch's sibling tool, if you will, called BCRMatch, which is under development. Not yet available, but you get to see the exciting work that's being done behind the scenes. So with that, Raphael, thank you so much. And this is TCRMatch.
I'll be giving the presentation on TCRMatch, so back to T cells. But before I tell you about TCRMatch, the tool, let's begin with a bit of a biological background on T cell receptors. Now, you've seen these figures here during the workshop today, and in the presentations about peptides binding to MHC class 1 and MHC class 2. Now, in this presentation, I want to focus on this part here, the part where the T cell receptor, or TCR, recognizes the presented peptide as an epitope. So let's use a different figure that details the TCR a bit more. This is the T cell receptor. It's a heterodimer where the alpha chain is encoded by a series of genes. So those genes that undergo somatic recombination before they encode for the protein. So here's a segment called a variable segment represented by letter V. Here's the joint segment represented by J, and here's the constant segment represented by C. In the second subunit of the TCR is the beta chain, which is encoded by certain gene segments, just like the alpha chain. Like in this case of the beta subunit, we have the variable, the joint, and the constant segments, just like the alpha chain. But you also have another segment called the diversity segment, and both alpha and beta chain contain three loops called the complementarity determining regions that are encoded here. I'm showing you the beta chain. So in the case of the beta chain, they're encoded by the BTJ gene segments. And so you have three loops that are called CDR1, CDR2, and CDR3 of the beta chain encoded in this BTJ region. The CDR1 and 2 are encoded in the V region, and the CDR3, as you can see here in this figure, it's encoded by the encoding span from the V segment, the D segment, to the J segment. So this, I'm focusing on the CDR3 beta because it's generally considered to be the most important because it is the part that most directly interacts with the peptide that is presented. So because the CDR3 beta interacts with the peptide, it became the focus of most research. And one technique that has become very common in T cell biology is called repertoire sequencing, which is the sequencing of the TCR population of T cells. So, in analyzing those sequences of the CDR3 beta is very interesting because it's a lot of diversity for a relatively small region. And it is small. Like these here are real examples of CDR3 beta sequences. They're usually flanked by cysteine and phenylalanine residues. Sometimes the phenylalanine changes to tryptophan. So now let's imagine that we have the sequences of CDR3 betas for population of T cells. That in itself tells nothing about the epitopes of our TCRs will bind to. So how do we find out which peptides our TCRs will bind to? There is one online database that contains curated T cell epitopes for us to use, the IEDB website, which contains sequences of CDR3 betas and their associated epitopes. So we can search the IEDB for the TCR that matches the ones that we have and see the corresponding epitopes. So it's a matter of taking each sequence here that was that we obtained and see, and each one of them has one epitope or more, and we can search for the epitope. And once the sequence is found there on the IEDB, you have the epitope for it. Now, in this example, with this sequence that I showed you, we were lucky. We had the exact match. But what about this sequence? What if we don't have the exact match? So I would still like to know which epitope it binds to. So how can we do that? We can use sequence similarity to infer which CDR3 beta contained in the IEDB is the most similar to our query, using, for instance, this amino acid substitution matrix. I'm showing you the well-known BLOSSOM 62. So our problem is to find the most similar sequence to our query. And the question then becomes, how do we compare sequences? Which algorithm do we use? Our team tested many algorithms, and I'm going to show you one graph from the paper, from the TCRMatch paper, that I think neatly summarizes the result. It's a graph of the precision recall curve. So if you've never seen this graph, the way you read this graph is the best result is to the right of the graph, and upwards is also better. So when they tested all of these algorithms here for sequence comparison, the one that best captured the right CDR3 beta in epitope pairs was this. And this one that I'm highlighting here for you is the algorithm that later became known as TCRMatch. Now, how does it work? So this table here at the bottom is from the paper. And it says it performs a comprehensive comparison of all possible k-mers using BLOSSOM 62 observed frequency matrix. So translation, it's a k-mer approach. First of all, we need the BLOSSOM 62 matrix because that's what TCRMatch uses to compare the sequences. So we have our two sequences here, the one that is our query and another one that is a putative, that is found on the IEDB website that we have a known epitope for. So how similar are these CDRs according to the k-mer method? So we're going to start with a k equal 1, and we're going to gradually increase this value. So k equals 1 means we take one amino acid, start from left to right. So the entry for AA in our BLOSSOM 62 matrix is AA gives us a +4. Now we take the bottom sequence and we shift one amino acid to the right, and we get A and S. So that's a +1. Shift right, A and S again, which is a +1 again, and A and I is a -1, and so on till we reach the end of the sequence at the bottom with AY comparison, which gives us a -2. Now we go to the second amino acid, the one at the top sequence, and repeat the procedure. So we have S and A, and the whole thing is all over again. So S and A gives us +4, and we repeat the whole thing till we get the end of the first sequence and the end of the second sequence, and we're done with k equals 1. So now we go from k equals 1 to k equals 2. And now we're going to compare each of the dimers. So we'll start with AS matching AS. So A and A +4, S and S +4. Shift the dimer at the bottom to the right. And then we have A and S and S. Shift by 1 again. A and S and I. Shift. You got the idea, right? Till the end of the second sequence. And then we shift the first sequence to the right, and we start the process. And this process for k equals 2 ends when we reach the final two dimers. And then we switch from K2 to K3 and repeat the procedure. And for K4, K5, all the way in this case for k equals 10. In this case, in the case of k equals 10, we only have two peptides to compare these two here. And in this case, in this example, it's k equals 10 because it's the size of the smallest sequence. And the final score is going to be the sum of all these values, all these entries that I went through in the example, summed. But then to give you a standardized output, we normalize it between 0 and 1. And this is what you get when you use our website. Let me demonstrate how we use the website. So we have here these 10 sequences in I believe there are 10 sequences in a text file. I'm going to copy them. And I'm going to go to tools.iedb.org TCRMatch. And this is our TCRMatch website. Then we have here a box that says enter CDR3 data sequences in CASTA, new line or space format, separated format, which is exactly what we have, a new line separated format. So pasting the sequences here. And we have here, or upload a CDR3 data sequence from a file. So we, instead of pasting, we could have uploaded this file here directly. But we don't have a lot of sequences, so just pasting works fine. Okay, next option here, we have some output options. The first one reads trim sequences. And it's pre-selected for you because we recommend you to trim the sequence. It means removing those flanking residues. Remember the F and the C, those flanking residues, we recommend you to remove them because they don't impact on the binding. So here I have another one. So the TCRMatch is going to automatically remove those. And next, you can select the database. So you can search the matching CDRs in the IEDB database using the entire IEDB database, which is this one that you've been seeing following since day one of the workshop, or you can use the alternative. If you're interested only in cancer, you can search on the CDR database. I think I'm gonna do this example using IEDB database. There you go. And next, you can select the filtering level. Remember that score that ranges from 0 to 1 that we just did the example with the blossom? We usually recommend you to use a very stringent criteria, so only those matches that are higher than 0.97, but you can use medium or low. Let's go with the recommended for this example, or show all the matches, yeah. So let's just go with the stringent criteria, and we also recommend, because sometimes it may take a long time, so you can leave your email address. So let's click submit here, and there you go. And there you go. So here we have an echo of your input parameters. And here at the bottom, you can download the results for in case you want to open on your favorite spreadsheet program. And I'm going to show you all the entries, if they're not that many. And we have here the output for TCRMatch, which I'm going to show you. We have the input sequence, the matching sequence. I'm going to show you this on the slide. I think it's better. So let's see here. So as I showed you on the website, if you run TCRMatch, this is the output that you get. So here in the first column, you have the CDRs you submitted. Here you have the CDRs for the that are in the IEDB database. And here the measure of similarity between them. So this is that score that varies from 0 to 1, as I explained. And this is the epitope that the matching sequence binds to. So if the matching score is sufficiently similar to your sequence, the epitope might bind to your sequence too. That's the idea. So there's also extra information here about the origin of the epitope, like antigen and source organism. But this is it. This is the standard output that you get when you input sequences, that you input amino acid sequences. Now, recently, we also adapted TCRMatch to directly take input from another program called Trust4. Trust4 is a program, it's actually a bunch of programs, a suite of programs, that says it's for reconstructing the clonal architecture of TCR repertoires from high-throughput sequencing data. So the main idea is if a bunch of T cells express the same TCR, then probably means they originated from the same ancestral T cell that underwent clonal expression. So the input is a bunch of TCR sequence data, and then Trust4 will cluster them and identify TCR clones. So if you download Trust4, and if you follow the instructions on how to run it, and I ran it, you're going to get a bunch of files as output. So I'm going to open one of those files in graphical interface. so we can see what we're dealing with. So the first thing I'm going to do is highlight that there is, here we have the nucleotide sequences for the TCRs that we used as input. And I'm going to remove this column because the file is very large and I want to fit the rest of the file in this slide. So what we're really looking for is this one here, the column that says CDR3AA, which is the amino acid sequence of the CDR3 of the TCR. So in the TrustScore output, there are many out-of-frame values, which means non-functional TCRs related to frame shift subcodons. So, and we have the sequences here, the CDR3s of our TCRs, which are the actual amino acid sequences that will feed into TCRMatch. So let me demonstrate how to use the TrustScore output with the TCRMatch using the website. I'm going to take the TrustScore input that I just showed you. That one, as you can see here, we have the counts, the frequency, the nucleotide sequences. You can upload the entire file. You don't have to copy paste anything. Just standard output of TrustScore. You can just go to the TCRMatch website. So it's tools.iedb or TCRMatch, and we have here the option upload TrustScore input from a file. So I'm going to take this file and upload it here. And I'm going to use load this time and submit. And this time it says the result table is too large to be displayed. Well, we have many results then. So it doesn't display here. We're going to have to download the result and open on Excel. So let's do that. The whole thing is here. As you can see, the output of TCRMatch now is the following. Here to the left, you have the input from TrustScore. And here to the right, you have the output from TCRMatch that has been appended to the input of TrustScore. And I have all the information in one spreadsheet. This is because I used, if I had, the result table is too large because of the options that I used. If I had chosen the CDAR database to search, then you would have less data, so the whole thing would have been displayed here. So yeah, let me show you here. This is the table, but the whole thing is in there, like the same thing. To the right, you have the input from TrustScore. Let me show you everything. Here you go. And to the left, excuse me, to the left, you have the input from TrustScore, and to the right, you have the output from TCRMatch. Let's see. And this is my habitual final slide. This is the part where I usually say that TCRMatch is the joint effort of all these people here. And if you're interested in how it works and reading the paper, this is the paper. But this year, there's one extra thing we'd like to show you. It's a new tool in development, but I will pass it to Mahita so she can talk about it.
Hello everyone. I'm going to be sharing my screen. Okay, I hope you can see my screen. So I'm going to be talking about a new tool called BCRMatch, which is a web server to predict antibodies that bind to a common epitope. And I'm just going to give overview on how it works. So this is like a mock-up of how the web server is going to look. And in the first step, the prevent the sequences are the 60 leaps antibodies without the known epitope. And so the antibody is epitope you want to know is referred to as an antibody of interest. And then in the second step, the user has the option to select modular models, and we provided 5 modular models at the training data set to make the prediction. So here we have 2 training data sets. And then the internet step, the server uses the selected pre-trained models to predict what antibodies share the same epitope with the antibody of interest. And then the third is prediction is rooted in a demo. And then we also recommend using a consensus of the prediction set of final learning models to guide the final result. So here we have shown only 2 learning models, but in the final we will provide a final learning methods. So that's the overview of the new tool. And thank you for your time.
Awesome. Thank you so much, Mahita, for this brief introduction to BCRMatch, and Raphael for the overview of TCRMatch. We hope this time next year, BCRMatch will be up and running and we can show you some live demonstrations as well, rather than just mock-ups. So that's awesome. We're running about 5-ish minutes ahead of schedule, so we have a little bit of extra time for the Q&A. So I can see we do have quite a few questions coming through. So now I would like to invite Eve, Raphael, and Mahita back to the virtual stage to complete our last Q&A of day 2. So Eve, did you want to take the first question?
Yes, very happy to. So I think the first question is from Emma Ryan, asking, is there no way to evaluate epitopes made from dimers, or is that planned? So you can use the current tools to do that by going in and modifying your PDB file so that all of the entries have the same chain ID. So there are a number of tools online, I think, that are available to do that, if not manually. And that's currently the workaround. If that's something that is that you would be interested in, not having to do that, that's good feedback that we can potentially implement. So thank you for your question.
I'll take the next one then. Lana Kelly asked, I may have missed this, but how does MHC factor into the data of TCRMatch? The Excel file did not list MHC data. Do then plot the epitopes of the highest matches? No, MHC does not factor into this. Like there's no, you can yourself come up with a pipeline that uses MHC prediction and TCR and the TCR side of it, but TCRMatch does not take into account MHC, like the whole MHC prediction or data.
So we've got a question from Shahzad asking which linear epitope prediction method is more reliable, ELIPRO or BEPIPRED, because they show some differences. So firstly, so ELIPRO is actually a structure based method, so I suppose it does, but you're right, it does also do the linear epitope prediction. So when we talk about reliability, often we're referring to their performance in specific benchmarks. And so generally speaking, I think ELIPRO does outperform BEPIPRED 2.0. When there's an update to BEPIPRED 3.0, actually BEPIPRED 3.0 outperforms ELIPRO in most benchmarks. But so if you're, I would recommend probably using ELIPRO for the time being as the more standard answer, but I think it's worth looking at both. Thanks.
I'll take the next one then. For TCRMatch, is there anonymous? For TCRMatch, is there an easy way to get gene names or identify matching antigen? The key to answer your question, I'm gonna say no, it's easy. There's no easy way, it's not directly in there, so there's no easy way, but it's not hard if the matching antigen, you can search for genes for the gene name, but there's no, it's not implemented on TCRMatch, getting it directly from there.
Okay, so I think so. I'll answer another question. So we have a question from an anonymous attendee saying that the B-cell epitope prediction tools are known for their low accuracy. What should we pay attention to when using these methods? What kind of applications should we use it in, and what applications should we avoid using it in? How should we interpret the results, and how much should we rely on them? So I guess there's a lot to unpack in that set of questions, and I probably don't feel comfortable giving hard and fast answers as to which specific applications you should avoid using them in. I think it's best to think specifically about what kind of error rate you will accept in your own application, and look at these methods in terms of their performance in the ROC curve, look at the specificity and sensitivity values at different thresholds, and think about how comfortable you are with using those for your specific application. In terms of interpretation of the results, I think that there's probably some value in using consensuses of methods. So sorry, it's a very wishy-washy answer for you there. It's, I think, a question of your application, a question also of your antigen. So there are some antigens that will be very well represented in the training sets for these methods. So if you're working with SARS-CoV-2 spike, that's very well represented in the most recent training sets for these classes of methods. If you have a much more out of distribution or unusual antigen, so something like malarial CSP with lots of repeats, then maybe you have to accept that the model is less likely to generalize to your methods. So yeah, sorry for the wishy-washy answer, but thank you, because we have to be wary of the limitations of these tools. Thank you.
I'll take the next one here. Does TCRMatch accept CDR3 alpha or combination beta plus alpha? No. As it currently stands, it does not. It only uses CDR3 beta. So if you input anything else that comes from alpha there, it's going to search a database of CDR3 beta. So, but this is in the horizon. It's in the makings. It's of course a research question, how to combine the information from alpha and beta in other CDRs. And but the current state is the one that we tested many times. So to deploy the versions in development, we have to test it and make sure it's all working. So, but it's in the makings. Thank you for asking.
Okay, so for the next question, I'm going to sort of group two questions. They have similar answers. So the first question from Manikanta Bondada about is it good to identify the B-cell epitope separately or within the context of interferon gamma inducing epitopes from MHC2, which method would be best to do? And then the second question, again, about the T-cell side of things, about differentiating between Th1 and Th2 related responses where the epitope is more associated with IgG or IgE. So unfortunately, these methods are basically trained on a high confidence clean set of antibody antigen complexes from the PDB. So there's very little information there about the broader immunological context in which those were raised, sometimes you have isotype information of IgG and IgE, but that's not easily extractable from the training data, nor can, unfortunately, that be something that you can get out from the B-cell prediction models. And yeah, in terms of predicting B cell epitope separately, or if there is some synergy with the T cell epitope prediction. I think that there is. I've seen, I think, one paper that has done some benchmarking of a similar thing, but I think there's not a clear answer to that yet. So yeah, thank you for those questions.
I think the next one, sorry, it went away. So what's the score normalization used? Sigmoid, max, something else? Because how exactly scores derived from multiple k-mers are compared? So it's neither of these, it's something else. It's this algorithm, the k-mer algorithm for TCRMatch, what you do is you use this algorithm on a self-match. You have two sequences. You run the self-match on sequence one, and then you run the self-match on sequence 2, multiply them and take the square root. And that's your normalization factor. And you divide by this number. And that's how you get the 0 between 1 that is standard, that you can use to compare or across multiple runs of TCRMatch.
Great, thank you. Then I'm just going to answer a question from an anonymous attendee about questioning whether there's a continuous benchmarking effort for the B cell epitope similar to the MHC prediction. So we currently do not have a continuous benchmarking effort for this. This is, I think, a great idea, and yeah, something that I think would be very valuable, because benchmarks can really differ from one another quite a lot. So yeah, thank you for that. Thank you for that question. Good idea.
So the next one here is another question from Chris Thorpe about including CDR3 alpha in the future versions of TCRMatch. Yes, we intend to include it. Thank you.
And I think we've still got a few questions. So we have a question from an anonymous attendee about how one selection for the prediction model can be done for each run. Is it possible to do pipelining to automate comparison of the results of different models? And I think that would be a great thing to incorporate in the next gen tools. I think that would be yeah, a real advantage of the pipelining. And I should also say, yeah, that lots of these are the linear B cell epitope prediction tools, for example, are available as a standalone. So if you're comfortable to do so, certainly the code is available there for you to, you know, to have a look at that yourself as well. Thank you.
I think I'm out of questions, Luke. So...
Okay, so I think we have a couple left that here. So I think Mahita's typing an answer to one. We have a question about in identifying antigenic and immunogenic epitopes, is it recommended to use the whole protein target sequence or in using the antigenic portion of the sequence? So I'm not sure I entirely understand, but I would say that it's probably best to include the whole protein target sequence as you would expect it to be recognized by the immune system, because there may be things there about if you include only a subset of the sequence, you know, that could be non-surface accessible, or so it's good to include the whole protein sequence. And then we had a question about from R Bussy saying, if I want to use ELAPRO for fusion proteins, I may have two different structures in the PDB, one for the tag and another for my protein. How should you proceed? There's, I know you can, there are a number of tools, I think, where you can combine different polymer entities from the PDB into a single file. But you would, of course, also want to make sure that they were in the same coordinate space and aligned properly. So that, I think, that probably requires some more thought and perhaps some more computation than would be trivial. So yeah, that would be my recommendation in that case. And then a final question about testing performance for the prediction tools. And I would, recommend having a look at the references. This is often sets of benchmarks. so sets of structures that everyone makes predictions on, and then you can compare. And that's sort of what the performance testing is. Thank you. Well, actually, I don't think that was for me, because that was after my talk. But anyway, thank you.
Alrighty, our last two questions have come in. Sorry, while we're clearing those out. So maybe, I don't know if it's for Eve or for Raphael, but while you guys look at that, I'm going to just repeat out loud from an anonymous attendee. The question was, is there a database of BCRs, sequence or structure that exists in healthy human under physiological conditions? And Mahita very kindly responded via text, saying there are databases of BCR sequences and structures such as our own, the IEDB, observed antibody space, structural antibody database that contain information about antibody sequences and structures. However, they cover a wide range of antibodies across diseases and not just quote unquote healthy individuals. So thanks, Mahita, for responding to that as well. So of the last couple, Raphael or Eve, anything else you'd like to add? Alrighty, I think we'll have to respond to the remaining via written response is probably the easiest thing. So with that, I will say a big thank you to Eve, Raphael, and Mahita for those presentations and answering the questions as well. Thank you all.